city?
No.
We're just the area. I know what Lexus is, some people say.
Yeah.
Just not even gonna spam from BME. Very quiet. Blended in with the students. So. All right, so we'll go ahead and get started. So welcome. Today is the first day of our Heart Month lecture series, which is now in its 7th year. So it's been going on for quite a while. And this is the first of 3 lectures that are focused on heart and vascular research. Just to note that the series culminates in our Heart Day Symposium, which will be on February 16th, this year over in Parkview Hall on MMC. So please join us all day that day. We'll have a lecture in the morning, we'll have a poster symposium, we'll have lunch and breakfast if you want to come early. There'll be an outreach event, and then there'll be flash talks by some of your peers in the afternoon. So it'll be a full day from about 8:30 till 4 o'clock or so in the afternoon. Today we're honored to host Dr. Stefania Nicoli, who joins us from Yale School of Medicine. She is an associate professor of cardiovascular medicine and genetics at Yale, right?
Yes.
Okay. And she directs the Zebrafish Phenotyping Core for Precision Medicine and is the co-director for the Yale Cardiovascular Research Center. She joined the faculty at Yale after completing a postdoctoral fellowship at the UMass Medical School and receiving the prestigious NIH K99R00 Pathway Dependence Award. Since joining Yale, she has developed a research program focused on understanding the function of RNA in cell and tissue development, homeostasis, and disease, with particular interest in cardiovascular and neuronal mechanisms. And in recognition of her work, she received the Judah Folkman Award in Vascular Biology at the North American Vascular Biology Organization meeting in 2022. So we are very fortunate and lucky to have her here today.
Thank you very much. It is a privilege for me to be here. Can anybody in the back of the room hear me? I'm sorry, sometimes my voice is great. All right, so yes, as just introduced, I am passionate about cardiovascular development and particularly mechanisms that control how cells communicate with the environment. So today, actually, shameless, I'm going to plug in a search for a PhD student and postdoc to work in the project that I'm going to show you, which is really one of the most out-of-the-box idea we have had in the lab. I would love to hear how the bioengineering, engineering mind in this room think about this problem. So a challenge for you to pay attention. So just as a brief and very basic introduction, we are really passionate about messenger mRNA. And this is a very oversimplified schematics of how messenger mRNA is being transcribed, occupied by a ribosome, which then will be important to make protein. And really, in the world of cell biology, developmental biology, many of us don't really think about mechanisms per se. We think often the amount of mRNA that is being produced equal to the amount of protein that the cell needs to actually function. However, over 60% of mRNA in a cell have microRNA binding sites. So what are microRNA? MicroRNA are essentially repressor. They bind the TRUTR or messenger mRNA, and essentially what they're there to do is correct mistake. So nature actually makes 60% of the time the error to produce too much mRNA than what is needed to have a correct protein, which is kind of a weird thing to think evolution allows such a big mistake. But nevertheless, that's why microRNA have been invented by nature so that to repress this excessive mRNA and therefore have a balanced mRNA to protein ratio. So for the past 10 years, my lab actually have been investigating this process, and we started with a very basic experiment, knockout microRNA in zebrafish, in cell culture, you name it, and understand what are the consequences of performing this type of manipulation. So briefly, we found, well, first of all, that if you do this mechanism to remove microRNA in a zebrafish embryo, for example, we found out that animals are perfectly normal, but they don't like to be in different environmental conditions, such as they don't like to be stressed, they don't like to be high temperature, having heart diseases, and so on. So they are much sensitized to what's called microenvironment. If you do these, for example, in a stem cell context, the stem cells, which are very dependent on their environmental niche, they actually become highly variable. So they produce different type of blood stem cell lineages, they are confused, they proliferate too much, and so on. And finally, we also did this in a cell culture where we took endothelial cell, removed microRNA in different conditions, and we found out that endothelial cells become more sensitized to the mechanical environment, including the stiffening of the matrix. So 10-year works, every time we remove a microRNA, we found out that essentially we create an environment where the excess mRNA compare what we think about sensitization to the environment. So after that, I really wanted to understand what is the connection for excess mRNA in a cell and the sensitization to an environment. And that's where the big mystery started. All right, so we started from Q in the literature, and one thing that we know is that mRNA is actually localized in what we can potentially define mechanical competent localization in the cell. For example, we do know that some type of mRNA, such as RAD14, seem to be localized during the migration, in the migration front per se. And RAD14 is also localized in the filopodia or lamellopodia of cell. Well, we have a very outstanding example of beta-actin. Bob Singer many years ago discovered that mRNA is actually polarized towards where the cell needs to move. And then more recently, our example sophisticated in mouse, where, for example, in the intestine of the mouse, ApoB mRNA is in the apical surface, or whether the ribosome are in the basal. So again, suggesting that mRNA have a way to localize to what could be the interface between this cell and the next environmental stage, with whether it's matrix, whether it's, of course, absorbers and nutrient. So perhaps there's something about mRNA and environmental views as an environmental. So we decide, therefore, to understand whether cytosolic mRNA could play a role in cell function, particularly the localization of it. And not bioengineering background, so again, we try to a simple thought. So let's take subcellular localization in the cell that the function is literally to sense mechanical environment. And to the best of our knowledge, one of the obvious, and also I work with Marty Schwartz, which is a very focal adhesion-oriented person, and he told me, well, look at focal adhesion, because focal adhesion, as you can see by these colorful image, are the sentinel constant sense environment within the cell. They form, they reform every time the cell needs to understand, and I'm going to the left, I'm going to the right. They sense the matrix. And they are actually incredibly complex. This is just a staining of a cell where focal adhesion and are tightly connected to the actin. But in a simplified and schematic view, focal adhesion are literally sensing the environment because they are connected to the environment itself. And by environment here is talking predominantly about extracellular matrix. And they therefore transduce signaling inside in the cytoplasmic compartment, and they perform a variety of cell functions, cell shapes, cell motility, apoptosis, differentiation, you name it. So something I also had to disclose you, that focal adhesion are composed of over 300 proteins. So per se, a focal adhesion is not an organelle, so to speak, so it's not like the ER, the mitochondria. But 300 protein is quite a big macromolecular complex. So if you remember the movie that I showed you, imagine 300 protein and once every few seconds they have to form and reform and constantly interchange among each other. Incredibly complex macromolecular protein complex. Another important aspect that is interesting to know is that the focal adhesion are also heavily organized in disease strata. What does it mean? Why their complexity is also very much organized by having an integrin facing the plasma membrane, extracellular matrix, and penicillin, taurine, vinculin until the actin. So overall, we are talking about a complex but organized and extremely dynamic protein complex. So it suffices to say that anything that goes wrong in this mechanism, an alteration of either phosphorylation, protein missing, different interaction, is pretty much one of the high causative phenomena of fibrotic disease. And if you want to some example of fibrotic cardiovascular disease, you name it, hypertensive heart disease, thoracic aneurysm, cardiomyopathy, and so on. So actually, although these molecules are fairly well studied in the field, particularly cell biology, when we come to disease, we all know that they are important and causative disease, but there's such a complex mechanism to alter. There's really no available drugs or available tools to manipulate that because they're way, way too complicated. And you don't want to get rid of them, you don't want to increase them, you want just to modulate their functions in a disease context. So with this complexity, and that's how our project starts always, very complex question, we started to narrow down how do we figure it out whether perhaps mRNA is entangled with this complex as a way to control environment. We performed this experiment in which was actually created Claire Waterman lab, did it the first time, where a cell sitting in a dish can actually hydrodynamically and osmotically inflate it, and then this body of the cell can be removed, and because focal adhesion are such tightly connected to the extracellular matrix, they will actually stay into their dish, and therefore you can collect what's called focal adhesion isolates, and then we perform what we like to do in the lab, it's omics, including isolate mRNA sequence that perhaps are a focal adhesion within ribosome protected fragment to understand which mRNA were bounded by ribosome as well as the protein. So first of all, here's how the experiment looked like. And we did this in an endothelial cell, but we also did this in human dermatofibroblasts, so two adherent cell type, which is very important related to the focal adhesion function. So the focal adhesion are, in this case, vinculin in magenta, and you can see that in the Western blot validation are indeed enriched in what is called the washout of the cell. And then we have actin bundle also at focal adhesion, but mainly every other organelle, such as the ER, the nuclei, and the mitochondria were washed out. So therefore, we suspect the preparation was actually correct. So the first experiment that we did is analyze, is there mRNA at focal adhesion? And the answer of that there is, and this is a Volcano plot of mRNA that is enriched in the focal adhesion prep versus everything else, and it was done both in UVEC and Humaderma fibroblasts. So the mRNA at this focal adhesion is about 90 to 100 species of mRNA, and we were able therefore to attempt validation of that by doing a technique that is called single molecule FISH. So the single molecule FISH is essentially an hybridation in situ where you have a probe that is fluorescently labeled, and it will make Watson-Crick base pairing with the endogenous mRNA. So a couple of things in this slide. So first of all, we use a control mRNA. By control means essentially an mRNA that was not found in focal adhesion. And the second one example of mRNA, TRAC2, that was found at focal adhesion. And you can see essentially that their distribution of mRNA is fairly randomic in the cell. However, if we overpose the focal adhesion staining, we can now observe how they have the tendency at least twice as much compared to the control to being enriched at the focal adhesion. So in this left panel, or the right panel to me, actually we tested what's called positive control. We didn't really know besides beta-actin if there was any other RNA rich in focal adhesion. And two things, beta-actin is so highly expressing everywhere, but indeed, if we look at the focal adhesion, mRNA is also present at this stage. So to us, it looked like perhaps we are into something. So there's not just beta-actin at focal adhesion mRNA, but there is an entire population of them, which could be interesting to continue investigating. So we were not 100% convinced because we only have a few targets to validate, so we decided to validate them all. And it's possible to do this with a technology that is called multiplexer single molecule FISH. So in this case, you color code a probe with a barcode, and then each barcode will match to a specific mRNA. So you can visualize in a cell 100, if not 1,000 of different single molecule species. And then in top of that, by localizing the mRNA, we also stain the cell for vaccillin and actinium-1, which is another marker for more mature focal adhesion. So you're going to end up to have an image that look like this, where the RNAs are all color-coded and focal adhesion are in white. And the more important thing for us is that we actually did validate it. I'm sorry if I point out, but I cannot really use the mouse. It's like weird. If you want me to stop moving, let me know. But nevertheless, the slide really suggested that 90% of the mRNA that were found by bulk RNA sequencing were indeed enriched at focal adhesion versus our control. And again, the control here, they were designed over 100 species of mRNA. They were either not found in our focal adhesion prep, or they were localized mRNA, but perhaps in other compartments, including mitochondria, ER, and so on. So we felt very confident that this was indeed suggesting that mechanical competent such as focal adhesion organelle have mRNA, which is kind of bizarre because mRNA is most of the time found in cytoplasm, while focal adhesion in this case are in the interface between the matrix and the plasma membrane. So next we did an experiment that I always had to make a premise of it because it's quite complicated. I have hard time here to explain it in the papers, but here it goes. Okay, so you might be familiar with single-cell sequencing. Single-cell sequencing, an mRNA that is expressed in the cell defines the cell function. In this case, we took the mRNA that were near a focal adhesion, and we decide what is the characteristic of the focal adhesion itself. And by doing this analysis, we actually found the mRNA are nearby 4 types of focal adhesion. There are mainly difference between their ability to associate with actinin, which is a maturation protein, versus the ability to be, for example, not associated with actinin and very smaller. Remember the picture about the focal adhesion being larger, smaller in the movie. That's exactly what it means, that focal adhesion associate to different type of focal adhesion based on their maturation cycle. So, and this is just a plot to suggest that track 2 prefer to be nearby a larger adhesion, T1 positive, and DCA2, for example, the opposite. So now you have RNA that not only localize the focal adhesion, but they seem to be localized depending on what the focal adhesion is functioning. So beta-actin is the only positive control that we had, and we know that beta-actin is this localized focal adhesion. This is actually one of the Singer paper because it's bound by ribosome. So the function of B there is to make a protein, which basically that's the main function of the mRNA. However, to our surprise, focal adhesion mRNA are not making the protein that then are the building block for focal adhesion. There's about 300 proteins, so there's a ton of protein, but only 195% to 2.54% of the focal adhesion mRNA are actually having the protein of focal adhesion. And this is just a representation that the focal adhesion mRNA have actually poor ribosome coverage. So in essence, not only they're not making the protein, but they're not very highly covered by ribosome. There's a way to validate that by performing a technique that's called PuroPLA, in which you visualize the nascent peptide of a protein. And some proteins, such as taline and vinculin, which are known to be focal adhesion protein, have a nascent peptide nearby the mRNA at focal adhesion, while the one that we discovered essentially had the mRNA, but they don't have a protein nor a peptide nearby. All right, a lot to take in, but I have break-ins. So the first summary is that there are a population of mRNA that is enriched in focal adhesion, are essentially differentially localized at different focal adhesion, and it doesn't seem to be contribute to the protein that are important in focal adhesion. Now, so first of all, how they can be enriched in that way, and we get also to the why. mRNA are not supposed to be there without making protein. Well, the mRNA field is an incredible and exciting moment of booming. Of course, mRNA is being mainly characterized by the fact that it's being able to bind RNA binding protein, and RNA binding protein are important for the RNA processing, but it's also true the opposite, that RNA is actually able to change the RNA binding protein function. RNA is also important to form macromolecular complex in what's called phase separation. So there are examples in which RNA per se can function independently for making protein. But what is important that you identify an RNA binding protein for doing that. And this is how we did this screen to identify this RNA binding protein. So we have essentially repeat the experiment before, and then we have removed RNA with RNase or cyclohexamide the ability of RNA to bind focal adhesion or to be enriched in focal adhesion. And when we screen for an RNA binding protein, they were lost at focal adhesion. So RNA is the glue that keeps the RNA binding protein at focal adhesion. So we found a few of them, and we did a small screening by shRNA, and it was immediately obvious that one of them, G3PP1, it was actually required. So in essence, when G3PP1 is lost, you can see that the RNA is now retracted for the focal adhesion, while the other RNA binding protein that we identified did not have the same phenomena, although they likely interact with each other, suggesting that perhaps G3PP1 has this ability to essentially crowd in the mRNA focal adhesion. We also validate the same idea by doing merfish in cell HDF lacking G3PP1. And as you can see from the control, there was a profound shift of this cloud of RNA that now are moving inside the cytoplasm and releasing from the focal adhesion. So G3PP1 is actually a protein that does itself interact with focal adhesion. This is a time lapse it will be fairly blobby, and I explain why it look blobby, because those are RNA complex, but you want to see a movie in which essentially there will be a formation of a new focal adhesion, and as soon as the focal adhesion forms, G3PP1 crowd around the focal adhesion, and it will stick with the focal adhesion towards the very, very end of their lifetime. So as soon as focal adhesion forms, this RNA binding protein, which recruit the mRNA, jump in and start to essentially allow a focal adhesion to form and reform. We also found that in the cell, not all focal adhesions have this G3PP1 RNA binding protein, but the one that do have, and it's about 50% of the time, they actually far more dynamics. So the one that have potentially this RNA, they form and reform in a much more dynamic way. We went ahead and understand how G3PP bind focal adhesion, and they do so especially via mass spectrometry and co-IP of G3PP1 by binding a variety of focal adhesion protein. And this is something important because the interaction between the RNA binding protein and focal adhesion is not actually a one protein, but many proteins on this strata that I show you actually have the ability to capture this protein itself. Finally, is this required for focal adhesion function? And I'm sure you're familiar, but just in case, the field is kind of skeptical removing proteins such as a binding protein with shRNA. So we did this experiment in which we essentially briefly remove G3PP1 by targeting G3PP1 with the dragon tag, so that when you add actually this compound auxin, the protein gets degraded in a matter of few hours. So then we test cell migration during this phenomenon, and we found that two important things. So the first one, there was a mild but significant defect in the way focal adhesion missing G3PP1 are able to cycle, and more importantly, the cell were not able to move. So that's a second part that I wanted to, we found a protein that is responsible to localize the RNA focal adhesion. We found that G3PP1 per se is required for focal adhesion function. So the final step is really to understand why. And this is like probably where the most obstacle we had, also in terms of reviewers, because an mRNA in itself in the cytoplasm is not supposed to exist if not to make protein. And indeed, when a cell is very sick, such as in condition of arsenide or some type of very nasty stress, hypotoxic stress, and so on, what happened that the ribosome drop the mRNA as a mechanism to start apoptosis, and there is a protein, interesting enough, G3PP1, that what does for living is form these granules, blobby granules, and the granules contain molecular mRNA. They are entangled with each other. It's almost a mechanism to protect the mRNA. In the meantime, the cell will deal with apoptosis, so that hopefully the cell can save itself. So in cytotoxic stress, there is a lot of information about how this protein controlling mRNA that's not supposed to make protein. However, we found that essentially these mRNA are naturally present in the cell. So mRNA in compartments such as focal adhesion are essentially potentially grouped in these G3PP granules. So these mRNA that we found in focal adhesion have actually some sequence characteristics that I'm going to show you briefly. So they are huge, they're much longer and much bigger in terms of QTR, as well as coding sequence compare the overall average of mRNA in a cell. They have a higher number of these called disordered domains, meaning that they love to make interaction with each other, and they also is reflected by the diversity. So they're not necessarily all RNA that can stay in focal adhesion. They have sequence properties that do that. So we felt like, let's look at potential granules that perhaps are forming nearby focal adhesion. And we did high resolution microscopy. And the first important difference is we did not see the big round granules that I just showed in the cytotoxic stress. So the granules that the RNA binding protein make with the mRNA are actually fairly diverse in their shape. And this is just a quantification. They are smaller, larger, and more in particular dependent on the RNA. But they are also more branched and a little bit less branched with other type of RNA. And this is just a quantification showing that every time there is a blob, there is an RNA. So the blob is actually specific, making the RNA be nearby a focal adhesion. So we wanted to understand why these granules are not dissimilar to the stress granules, which are rounded. The reason why we were curious about that, because there are biophysical properties of the granules depending on the shape. The rounded are likely to be more liquid-like, and the branched are more likely gel-like. And this is based on knowledge in the liquid phase separation field. So to understand a little bit better what is behind the property of RNA to make these different granules, we did this assay in which it's possible to reconstitute the granules in vitro. Now, I know that in vitro for you is cell, but actually this is a real tube in vitro, and that's a living cell. And what's interesting right here, that the RNA that we found at the same focal adhesion make the exact shape and branched that they make it actually in vivo. So the RNA sequence have a memory of what kind of shape of granules I need to make in a tube, and this was actually possible. Remember this weird data that I showed you? I'm pretty sure it was puzzling. Well, why different sequence RNA would be in one focal adhesion and the other in a different focal adhesion and so on? The reason is why, because they need to mix with each other. So we took mRNA that like to be at focal adhesion state 4, which is a mature focal adhesion, and we mix in green, we fluorescently label each RNA. This is in green, track 2 and KFC. And when we mix that, they still make these very branched granules. And this is a quantification of their free energy, which is essentially the ability to be very comfortable in a certain conformation. And the ability to form what's called RNA-RNA intermolecular interaction. As you can see, they love to be with each other when they're mixed. If I mix two RNA that were found in focal adhesion more immature, they love to be with each other. But if I mix two RNA that are one mature and one non-mature focal adhesion, they do not love to mix with each other. Now you can see their color really try to separate. So the logic to distribute RNA focal adhesion is all about in sequence, and it's all about the property to essentially form these very branched homogeneous interaction. So we went ahead and figured out, is this minimal? Is this sufficient to form the shape of these granules? And I'm sure you've done it in the past, but basically, most of the assay for sufficiency is done mutagenicity, found the sequence, tested. That's how we did it. We took one RNA as a proof of principle, we have many other, chop it randomically in 500 nucleotides, take each sequence and perform these branch assays. And we found indeed that the sequence that were forming these very branched elongated were indeed, and this is just the shape of the quantification, but they were indeed having a much lower free energy, suggesting that essentially the one that formed the branch, they likely prefer to interact with each other. So these RNA domain that we found in the blue circle right here, they look often like this. So it's actually possible to mutate them, and we did this. And we found that if you mutate the RNA-RNA interaction in this sequence, you essentially remove the ability to form the branched, but more the smaller one. So okay, well, thank you so much. I could care less if you don't tell me what is going on here. I tell you what's going on, and this is actually one of the very interesting experiments, okay? Now, if an RNA is able to function just based on the ability to interact with each other, so then who cares about coding or coding? Just take a sequence of RNA that is making interaction with themselves, and then a control sequence that does not make this RNA interaction. And then we did this experiment. This essentially is, it could be defined as an RNA nanoengineering experiment, just to be fancy, but it's actually where you take a focal adhesion, we took 2 of them, protein, you fuse them with an RNA binding motif. So now you make a protein able to bind RNA, and then you actually feed the cell with these RNA fragments that have the ability to be recruited then by focal adhesion protein. And you can do this, and you can reproduce what we see, this branched morphology. For example, the spheric one, the one that don't make branch, are 3 molecule RNA, while the branched, all the molecule RNA are inside this very, very larger sequence. So if the principle is these RNA are there to form large branches and G3PP1 granules, what's the property of a protein that looks like that? And this is where it's starting to be very interesting. So let's say we target actinin, which is one of the proteins that naturally bind G3PP1. I want to remind that. First of all, we saw a decrease of actinity in the recovery by using an assay that's called FRAP, which I'm assuming might be familiar. So basically, you kill the protein and you measure how much time it takes to the protein to reconstitute the focal adhesion. And having a branched RNA clearly impairs the mobility of actin. It also impairs the mobility slightly of paxillin, because again, in the strata, paxillin is actually dependent on the actin sequence. And if we do this cell, if you actually observe their function, we found that the branched sequence of this RNA were actually impairing severely the migration of the cells. So these cells, they don't have any signaling impairment. The only signaling they have is a small sequence RNA attached to the actin, and they are not able to move. Okay, so then we did a second experiment. So why not just one protein? Let's tag another one. So we target with the same RNA sequence, then paxillin, that it's a little bit closer to integrin and much more regulatory layer. And so actinin in this case, FRAP was not affected, and it's likely because of the hierarchy of the structure of the focal adhesion. But again, the branched diminished very significantly the ability of paxillin to move when it's trapped in this branched granule. Now, if you follow the behavior of the cell, now we can create a different pattern of behavior. If you can see, the one linked to the branch, they move way much faster, so they keep moving, and they move in a very chaotic way. So they're completely disorganized. So with that, I would like to conclude it with and discuss a bit of this data. First of all, we think that the focal adhesion are engulfed in this G3PP granule to tune migration behavior. So as soon as focal adhesion form, they will be able to recruit different species of RNA that change what we think the protein mobility of focal adhesion itself. Although the application of what we learned is potentially massive, we can potentially engineer the RNA multimerization, so the ability of these RNA to bind to each other, to essentially engineer the protein behavior and eventually the migration pattern. Because in the case of actinin, we can stop the migration of cell. In the case of actinin, we can actually promote it or eventually change the directionality of it. And I'd like to mention that all this behavior can be manipulated in multiple cell types. So the project that we have in mind and probably to continue investigate is the following. The field of RNA nanotechnology is exploding. With the discovery, for example, the Cas9, again, Cas9 RNA is just one example, but you can imagine how people think about RNA nowadays. RNA can be packed in so many conformation and shape, which is essentially what naturally probably G3PP1 is doing. It's taking this RNA, then while they're making the protein, they have a period of time of burst of transcription and then a period of time of basically pausing if they capture in this sequence. So what we want to investigate, how much can we push the cell to do what we want by engineering the RNA in this macromolecular complex. Also, we only look at focal adhesion, but imagine if this could be true for cell-cell junction. I don't know, you name it. I studied tyrosine kinase for many years with super incredibly complex kinase nearby, so I can only imagine how much cell behavior we can change. And finally, of course, the film nanomaterial is also expanding. So how do we get this RNA to do what we want is the challenge, because right now we only use these viral sequence MS2 and MCP to get them in, which is very difficult to think about therapeutically how to manage them. And with this, we really have the ambition. We just started mRNA therapy, and I know you're aware of many of them are actually focusing in cardiovascular disease. And what I would like to challenge and to think about behind translation, what can we do with RNA, particularly thinking about fibrotic cardiovascular disease, where focal adhesion are really tough to manipulate. So with that, I'd like to conclude and take all the questions that you might have in clarification. But let me finish by thanking the protagonists of this story, particularly Liana Boras, which is an exceptional postdoc, potentially looking for a job soon. And Lorraine and Peter are two PhD in the lab that have helped learn to pioneer. It's an extremely interdisciplinary project, as you can imagine. We have help from single molecule expert, we have help from RNA sequencing and RNA structural, as well as single molecule imaging. So that's the beauty of Yale as an environment where you have an idea and you found the perfect and probably the most experienced person to do that. So we are very fortunate that it was possible. And then of course the funding for, not funding this project, but funding our, this is a very out of the box project, but we feel like could potentially rewire how do we use it in the future. And then I'll take any question. We have plenty of time.
Questions? I'll start with one more. I have a couple, but let me start with bigger picture in terms of, do you see RNA conformation and or the species change when you look at some of the development, for example? I'm thinking about times when cells are very migratory, right, and change, or cancer metastasis, right? Is there something there that you know?
So we just starting to do that experiment. The first thing we did is to take this RNA binding protein GGP1 and make different mutants in zebrafish, which is our in vivo site of the lab. And we did found migratory behavior defect, particularly the migration of the angioblast in the midline. So very, very early on. And on top of that, it's not just specific for the endothelial cell, but also the cytogenesis was also affected. So there is clearly a migration defect that is related to whether it's linked to this property of RNA is something that we are investigating.
Have you tried? I don't know if that would have any difference from the last experiments you did, seeing the RNA and the protein interaction. Have you tried RNA immunoprecipitation, for example, to confirm the finding?
Yeah, it is supposed to, we can do that. Indeed, by bioaccumulating, there is some chemistry that it's possible to immunoprecipitate, and that could be something that would be interesting to do. It could also be interesting to think about how each protein in the strata may be necessarily proximate to different RNA. So our study right now are mainly not in a Z, because the turf microscopy and everything is not very deep. But one can think that different RNA may be brought at different time that the strata is forming. And one way it could be, again, to bioaccumulate the protein and do an RNA sequencing depending on which protein you bioaccumulate in the facility. So yeah, there is definitely, now that we know and we have the hypothesis, it would be probably a very interesting experiment.
Follow up on that. Oh, sorry.
No, you don't.
Well, just because then it might relate. How does it get there to start with, right? I feel like,
Yeah.
and I guess too, they are focal adhesion related mRNAs that are there, right? So it's not
Yeah.
just random mRNAs, they are actually mRNAs associated with the proteins that are
Right.
there as well.
Yeah, so the 2% of the mRNA that's making a protein, like the mRNA of taline is actually making the protein of taline there. So that's probably, it makes sense that it gets transported there. Whether the transport is like directional or not, we don't know. Honestly, when we took this live imaging of the RNA, we actually did also live imaging of single molecule RNA. We don't see a transport to focal adhesion in a directional way. So it doesn't appear that there is a logic or molecule that transport there. How do they get there? It could be they perhaps have sequence-specific domain. We didn't find any in the coding in 2TR, but maybe in the FIU TR, something that we are looking into it, that when you have a very long coding sequence and you hide the domain, they might be bringing by another RNA binding protein to focal adhesion. It could also be, although it sounds horrible and I don't think it's true, but it could be serendipity. So if you think about where are focal adhesions, so they are maybe in the lamellopodia, at least in a dish, right? So we don't know in vivo how that would function, but clearly they are in compartment with cytoplasm is actually compressed. So the likelihood to be near a larger RNA is higher compared being, in the nuclear and so on. So it might be something that the system doesn't plan, but it's almost like an immune system for mechanics. So when you have to quickly, you don't have the time to think which RNA, they just take whatever is available that had the sequence characteristics. So although it's pure speculation.
Great, like, I loved your talk about like a lot of cool imaging stuff that I've never seen before, so.
That's great, interesting.
I was wondering, you spoke about how you like classify the different maturation states of focal adhesions. Could you maybe just explain a little more,
Yeah.
like how they're different? Like, do they just grow in size or? how?
Yeah. Yeah, so so it's a very good question. So the focal adhesions are being classified as what's called native, which essentially are forming and disassembly very fast. Some of them, they will not be forming and mature to actually bind the cytoskeleton, but they're just there for the cell to put their head in. Why the mature one are the one that will build the strata and connect to the actin, then signaling, and mechanically essentially form traction so that the rest of the cell can move forward. So the mature focal adhesion versus the native seem to be the turnover required for cell movement. So that's why we think at the moment the cell movement is predominantly the main behavior that we found. We also tested the property of the matrix. So we cultured the cell in different matrix, protein, fibronectin, collagen 1, 4, and laminin. And clearly these adhesion are the one with the branched RNA. Essentially, they prefer to be in a more stiff environment rather than non. So there might be other behavior related to the more mature adhesion to recognize and stiff in a stiff environment, but it's early days to know.
Or are those associated, so you mentioned the fibronectin versus collagen, that's different integrants that are there as well. So is there
Yes,
integrin
indeed.
specificity?
Yeah, there is, definitely. Okay, well, that's a mystery of all of the integrins seem to be, I didn't try to relate integrin. It would be cool to see if I actually, like the main protein that does this, what happens. So by link, I mean attaching the mRNA to that. So yeah, right now we just play with the regulatory layer of the focal adhesion.
Other questions? Yeah.
I don't know if I'm gonna word this correctly, but do you know,
The more crazy and the better here, so we'll go for it.
do you know, more or less how well it is, like the RNA, how much does it contribute to actual cell function itself, generally speaking, or is everything else that's going on with the cell more?
Yeah, I don't know. I mean, you're thinking perhaps to do like a understanding what signaling are altered by this. It would be interesting. I do know by the RNA linking experiment. We don't interfere with proliferation. That's something that the only thing we tested. But again, as far as downstream signaling, how different is the cell with this RNA, we don't know. The other thing is that normally speaking, cells don't like to have RNA. In this way, you can imagine how viral RNA are actually doing these things. But of course, when you introduce a viral RNA sequence, you have this immuno response in a way that the machinery of the RNA inside the cell will kill the RNA right away, or will try to do that. While here, I think we bypass that phenomena because we are essentially using endogenous sequence, and therefore likely there is no yet a response. But I think one hypothesis behind how the cell states look like, so it's actually a normal cell, it would be also to look for these mechanisms that actually are there to prevent RNA to form this complex.
Thank you again for the talk. That was quite intense for me. At least I'm not a cell biologist, so I have tons of questions, but I'll break it into two.
Yeah.
My first question is, like, how unbiased was your approach at determining the mRNAs that are associated with the focus regions? And then have you tried looking at it from the other way, like instead of looking at what mRNAs are attached to the focal adhesion, like, what else are those mRNAs doing in other parts of the cell?
Right.
You showed very nicely that they do non-randomly bind and associate with the focal adhesions, but you also see them all around the cell with the RNA focal adhesions. What do you think of those mRNAs are doing?
Well, I think in the rest of the cell, they are making the protein. So in the cytoplasmic compartment, indeed the protein they are making is not like it's not expressed. It is expressed, not a focal adhesion. So likely when they are not nearby a focal adhesion, they are actually doing what it's supposed to do. So all these mRNA are making protein. They are indeed important for other things, not for focal adhesion, but for like ER biogenesis, nuclear complex, nuclear factor, and so on. So luckily that's what they're doing. How unbiased I was, I mean, it was basically, initially it was a very, so we took all the focal adhesion with RNA sequencing, which usually is more the screening pieces, that's how we start. Indeed, a probable challenge right now. So how do we move forward, right? So obviously not every organelle can be purified or isolated in a certain specific way. However, again, as I mentioned, the RNA non-engineering is exploding, and there is now 2 papers that just came out this month where they are actually screening almost a transcriptome analysis, but rather than just read the sequence of an RNA, they actually are able to develop technology to read the hybrids. So in essence, they can see and quantify how many times or where in the transcriptome, hopefully in the future, this RNA interaction form. So that will enable us now to interrogate not only the level of expression of an RNA, but also perhaps in the future the RNA interaction and organization. So it's really picking up quite a big field. So hopefully we can screen for that, not because just of the sequence, but the ability to form interaction.
One last question.
And anytime, we're still fine.
Do you think, like, for epigenetics could be contributing to this findings somehow? Like,
yeah, I will say so. Okay, so there's 2 answers. So the first one is that actually the RNA already known to contribute to epigenetics, so the way around. So in the nucleus where the nascent RNA that is being transcribed is naked, so there is no ribosome. There are already evidence suggesting that the RNA in itself could function as a glue or crowding agent for transcription factor to then promote or repress different type of epigenetic change. So I think that the RNA itself change epigenetics. Whether the epigenetic change the RNA on the cytoplasm, it could be, depending, again, it might be a guess, like, clearly the epigenetic probably is not thinking about the non-canonical function of the RNA, but based on the protein. I mean, that's what I'm speculating, so I need more protein, they make more mRNA. But that's how actually our question started, why the cell will transcribe more mRNA. You need it. and it's never less, it's always more, right? So, and I think obviously it could be a safekeeping, you just throw it away and then someone else will fix it. Or it could be that you can use RNA in multiple ways, therefore, even if you have an extra copy in certain localizations, actually beneficial. So it genetically might have been evolved to make more mRNA. But this is all theoretical, of course.
Do you see, you mentioned you don't see changes in proliferation when you change it, but do you see, like if a cell can't form to adhesions, it will undergo, I think they call it anoikos, right, where it's got apoptosis, where it can't sense this environment. Do you see that over time? Or do you...
I mean, with G351, the Dragon system, probably it will be the case because those cells really don't move without the RNA binding. With the RNA fragment, it's different. So the dynamics of the adhesion is different, but not that they can or cannot form. And also, for example, with the actinium link here, right? So I'm only modifying potentially the more mature one, but I'm leaving the native format. So we can potentially actually play with whatever protein that we are interested in to create cell behaviors. Because manipulating the turnover of the adhesion is quite difficult because they're either everywhere or they have multiple functions. So I think it would be interesting to throw a bunch of making like a library of this oligo and do a screening for cell behavior in different proteins. Yeah.
That's cool. Any final questions? If not, I'll remind you that graduate students, PhD students can have lunch with Dr. Nicoli at noon over in EC 2680. All right. Let's thank her again.